Yeah, so I'm going to go ahead and do the introduction. So hello, everyone, and welcome to today's Precisionary Instruments webinar. I'm Abby, and I'm really happy to be your host. It's my pleasure to introduce Dr. Virginia Puente. She is an MSCA and Maria Zambrano researcher at the University of the Basque Country. Oh, somebody wants to join our call. Dr. Puente studies how the brain's extracellular space shapes neuronal signaling using advanced tools like STED microscopy, optogenetics, and shadow imaging. Today, she'll share her methods for preparing and imaging organotypic hippocampal slices, including the use of roller drum culture and troubleshooting tips to optimize these experiments. So we are really thrilled to have her. And please join me in welcoming Dr. Virginia Puente. Thank you so much.
Thanks so much, Avi, for your introduction and for hosting me today. Thanks so much also to Precisionary Instrument for inviting us to give this talk. I'm Virginia. I'm so sorry that today the camera from my computer is not working perfectly fine, and I could not put my face on there, but I'm happy to present today. And as she mentioned, I'm Virginia. I'm working in the Faculty of Medicine at the University of the Basque Country and Biophysica Center. And the main project under my leadership focuses on modulating neuronal signaling by brain extracellular space structure and dynamics. And this project in live tissue. So I do work with live brain slices from mice. And this project also aims to characterize the diffusional properties of the extracellular space and investigate their role in modulating neuronal signaling as observed in spatially resolved microscopy images of live extracellular space. But what does extracellular space represent? So cells in the brain are surrounded by this space, which forms porous net and interconnected routes for molecule transportation. And our view of brain extracellular space has changed from a largely and static compartment to dynamic and diverse structures which actively modulate all the part of the brain. Emerging evidence indicates that the dysregulation of brain extracellular space contributes to the pathogenesis and development of neurological disorders, highlighting the importance of therapeutic modulators of extracellular space function. So my aim is to study the dysfunction of the brain, more specifically of the extracellular space in pathological and physiological brains, as well as to develop innovative tools to study the properties of the brain extracellular space, and to develop also modulators to manipulate the extracellular space in the brain. As mentioned, I do work with live slices, and more specifically, I do work with organotypic hippocampal slices. We do work with the animal facility at the University of the Basque Country, where they take care of the wellness of the animals. And we do the dissection of the brain of the mice of the brain and the slicing there too. We do work with organotypic slices because they keep the site. They keep in the case of the hippocampal slices, they keep the site architecture and the synaptic circuitries of the intact hippocampi. And in the case of the organotypic slices, when we work with them, we keep the maturation and plasticity of neuronal circuitries, which is really interesting because we could keep them over several weeks in vitro in this kind of incubator. But how we reach to get these slices? So for that, we use the ultra precise and consistent vibrating microtome. The one we have is the Cumberstone Vibratome VF510-0Z. And we do the slices of the hippocampi. And I will explain you how we make all the steps. So first of all, we prepare a 2% agar mold by casting molten agar into a dish and letting it solidify. When it is solidified, we shape the agar mold to fit the specimen tube and create two small holes in it so that we could later place the extracted hippocampi into these pre-made holes. This 2% agar mold, we glue it to the bottom of a specimen tube to keep it stable. We dissect the hippocampi and we place the extracted hippocampi, as mentioned, into pre-made holes in the agar mold. Just for you to know that Precisionary instrument provide two kind of a specimen tube with two different diameters for this compressed we have. The one we have is the one with 15.5 millimeters diameter, and it's more than enough for slicing the hippocampi. And some researchers from my team, they also use it for slicing the whole brain of the mice. Once we place the extracted hippocampi into the pre-made holes in the agar mold, we fill the remaining empty space around the hippocampi with 2% agarose. And we chill the specimen tube with the samples with a chilling block, which is also provided by Precisionary instrument. Once we have the specimen tube ready for being introduced into the compressed tome, we start slicing. And if anyone has any question about the settings we use specifically for slicing the hippocampi, we could happily share it, and any requirement that you could need. So once we finish the slicing, what we do is to take the hippocampi slices, and we start the part of the culture. So for that, we use some specific cover slip that we later on use them for the microscope, for the homemade microscope we have. And on top of the cover slip, we add 10 microliters of chicken plasma and 10 microliters of thrombin, and we put on that the slice. After that, we introduced after around 20 minutes of having this in the fridge, we take the cover slip and put it inside these tubes, which are roller heirloom tubes, which we put them inside a specific incubator we have for keeping those samples for over several weeks. This is so meaningful for us and useful for us because in that way we could have access to these samples during different days, and we don't need to sacrifice animals every time we need to do an experiment. So if anyone is also interested in this kind of protocol, we will be happy also to share it and answer any question you could have about it. And in order to map the extracellular space across these hippocampi slices, I do use a super resolution shadow imaging, which is a technique that enables to visualize the intricate extracellular space structure with nanoscale resolution. And for those ones who are not familiarized with SUSHI or shadow imaging, it's an innovative technique in which we use the non-cell permeable dye, like calcium, for example, which is its structure here. And here, with this shadow imaging technique, instead of the surrounding of the cells rather than the cells themselves are made to fluoresce. So you see cells like shadows on a fluorescent background. And when we combine shadow imaging with super resolution techniques like STED, we could get a detailed visualization of the neurophysiologic structure in brain slices, facilitating the deeper understanding of neuronal connectivity. For those ones who are not familiarized with super resolution STED microscopy, STED comes from stimulated emission depletion, and it's one of the techniques that make up super resolution microscopy. And here there is a selective deactivation of fluorophores. Then that makes to minimize the illumination area at the focal point, and that enhances the achievable resolution for a given system. For STED microscopy, it is needed a redshift STED laser, a depletion laser, which has a donut shape around the excitation laser spot. And with this depletion laser, what we do is to stimulate the fluorophores in the excited state to come to the ground state without emitting fluorescence. So that makes turns off fluorescence, except at the focal point. And you could shrunk the spot of the STED laser, and that makes that could reach we could reach a resolution bigger than the diffraction limit, and this is the way we work. So we have a homemade super resolution setup in which we used our samples, and I have designed different chambers so that our cover slips with hippocampal slices could be adapted to them, and we could process and acquire images. These are, for example, some images we acquired a few weeks ago by using super resolution shadow imaging, and these slices were sliced with a Comperstone from Precisionary instrument, and these are some organotypic hippocampal slices we kept in culture for a few weeks. Here in these images, we could see the neurons as in white, and the background it's all the extracellular space in black. Just for you to mention that on our homemade setup, we are also implementing optogenetics, and that's why I'm starting to test some construct. More specifically, I have started using optogenetic construct expressing channel opsins, more specifically the channel opsin 2, which is an enhanced version of the previous one, where there is it is improved the channel conductance, and it is stacked with mCherry, so that we could combine it with fluorophore that we normally use it for doing shadow imaging. And in this case, this construct is expressed on neurons because it has a promoter specifically designed for being expressed there. So we could selectively choose the neurons which are expressing this construct, and work with that cell, that neuron, and see the effect it could have on the extracellular space. So by activating these channel opsins, the idea is to study the extracellular space dynamics around individual activated cells by doing two-color SUSHI. In parallel, I'm also working on re-engineering a blue light switchable synthetic extracellular matrix protein that allows to reversibly and non-invasively control integrin-mediated cell adhesion. Here, the photo ECM, in this case, it has an RGT sequence, which is upon blue light exposed, and that induces the interaction between extracellular matrix and cells. So the idea is to have optogenetic control over mechanotransduction through interaction between cells and the extracellular matrix by using this photo ECM new version I'm developing. In parallel, I'm also testing on our organotypic hippocampal slices fluorophores, which has been recently designed for labeling the extracellular matrix, which is, as you could know, part of the extracellular space. So in this case, there is this newly synthesized fluorescent dye, which binds selectively to glycans, and it's a cell impermeable dye that turns on upon low affinity to these molecules. And this is an overview of the talk with all the sections I have talked about. You could stop me or ask me later all the questions you could have on each one. So here the first part is I talk about how we culture the organotypic hippocampal slices, how we slice them with the compressed tone from Precisionary instrument, with which we are really happy to use, and we have made a huge progress compared to the previous setup we were using, and it's making our life much easier. And then this is the homemade super resolution setup we have, where we are also using it for doing shadow imaging, and we have adapted the specific chambers for the slices we have in culture with the specific cover slip we used for it. And as mentioned on our homemade super resolution setup, we are also applying optogenetics and all the kind of tools that help us to study the extracellular space dynamics. And these are some of them. So with this finishing, I wanted to thank to part of the researchers who are also taking part on this project. Also the funding I received for carrying out this project, which is ongoing, and Abby for sure for hosting me and in Precisionary Instrument for inviting us to give this presentation, and to all of you for your attention. Thanks so much.
Oh, Dr. Puente, thank you so much, and for showing us how you your wonderful sections with our chromosome. I want to open up the floor and see if anybody would like to ask any Q&A. Please feel free to unmute yourself or show your video. You don't have to. I don't have any comments or questions here in the chat, but please don't be shy. Does anybody have a question or like to explore more? And if not, don't worry. Sometimes you think of questions later on. And if you do, please don't hesitate to reach out to us at Precisionary. You can reach me directly at abby@precisionary.com or our whole team at info@precisionary.com. So with that, Dr. Virginia Puente, I just want to say thank you so much for your talk and webinar today. And I hope everybody has a wonderful week. Take care.
Thank you, everyone.